Wendy, you can pull a crowd, right? Um, Good evening, colleagues, students, guests. It's a wonderful pleasure to welcome you this evening to this much anticipated inaugural lecture. My name is Musa Mushabela. I'm the Vice Chancellor here at the University of Cape Town. And tonight is the final lecture in the 17 lecture series for 2025. And the lecture will be delivered by Professor Wendy Burgers. Let's give her a round of applause. For any academic, an inaugural lecture represents a major milestone. It brings many years of research, teaching, supervision, and public service into one single moment. It also provides us with insight into the ideas, the questions, the commitments that have shaped our colleagues' work. We are all here to witness that. These are always very personal occasions also, because family and friends are also in the room. They are also important institutional moments for us because each lecture and the contribution from our colleague deepens the intellectual life of this university. A warm welcome to Professor Burgers' family, friends, and colleagues who are here this evening. Your presence will help me because it means that Professor Burgers is going to have to deliver this lecture in a way that I can also understand. But you also get to see the sum total of what she's been doing for many years. And we as the university recognize that the achievements that we see, we benefit from, rely very much on the encouragement and support outside of the laboratory, outside of the classroom, and outside of the office from all of you. So thank you very much for your contribution. Let's give them a round of applause. I'd also like to thank and introduce you to the members of the platform party. Firstly, the Dean of Health Sciences, Associate Professor Lionel Green Thompson. Welcome. He will be giving closing remarks. The Director of Outliers NPO will offer a vote of thanks. Welcome, Dr. Heidi Siegel, and welcome back to UCT. Let me introduce the Deputy Vice Chancellor for Teaching and Learning, Professor Brandon Collier-Reid. You are most welcome. Thank you for being here. Let me also introduce Professor Carolyn Williamson, the head of the Division of Medical Virology, who will soon introduce our inaugural lecturer. Thank you for being here. So tonight we'll be treated to a lecture titled In Defense of Us All. This is not about the military or the police. It's engineering immunity in the age of outbreaks, and it couldn't be more relevant. We live in a period where questions about health security, vaccine readiness, and the impact of infectious diseases sit at the center of our public debate. Many of us talk about pandemic preparedness and so forth. Many of us witnessed the contribution of much of this work during the COVID pandemic, and we continue to witness these contributions from Professor Burgers and colleagues. These questions, of course, shape policy choices, economic planning. I hear a lot of people these days talk about how health is the new wealth. They influence global partnerships and very much personal decisions at an individual level as well. They remind us that science is not just an abstract concept, it sits in the middle of society's most pressing challenges. So Professor Burgers' work contributes directly to this field. She has established a respected research career in virology, focusing on how T cells respond to viruses and how vaccines contribute to immune protection. Her leadership of the cellular immunology platform has strengthened our ability to test vaccines, analyze immune responses, and support biotechnology innovation. I have to say that I'm biased because I'm very much interested in that part. This platform has also created space for collaboration with partners, academic, scientific, but also in industry and private sector, including partners such as Afrigen and BioVac, thereby strengthening the local capacity necessary for long-term readiness. So as we wrap up for 2025 with this amazing lecture series and the lecture today by Professor Burgers, we are here, Prof, to witness your contribution, to celebrate your achievement, and to also support you and work with you in this journey of a broader purpose of tradition at this university, which is quite sacred to all of us. These lectures help explain the value of scholarship, and I'm sure today it's not going to be different. And they also demonstrate how the work that you do impacts the public life and enhances institutions, ours included, and expands the boundaries of knowledge. I want to thank all of you present here. There are many special guests in the room, and I'm very grateful that you've all managed to find the time to join us. And I also just want to thank all the students and academics and scientists that are here. Let us all congratulate Professor Burgers with a round of applause. Prof, your work reflects the best of what our academic community strives to offer: depth, clarity, purpose, and service. Congratulations to you. Let me now invite Professor Williamson to introduce our lecture. Thank you very much.
Thank you, BC. Good evening, colleagues, friends, family. I want to give a special welcome to Wendy, Wendy's mother, to this celebration, and her boyfriend, Kent, and then the Seagull family, who I'm told by Wendy is her extended second family. So welcome also to you. I also want to acknowledge and welcome, we have a few guests who've come in from WITS. I only saw two of them, Penny Moore and Glenda Gray, and so a special welcome to have you visit here as well. I'm sure there are probably others who have come from far away. So it's a great honor to give this introduction to you, Wendy. I've known you for 25 years. And during this time, I've got to really like you, respect you, and admire you. So I first met Wendy when she was in medical virology many years ago, pursuing her MSc. She performed really well during this time and was awarded a Commonwealth Trust Scholarship and an NRF prestigious overseas funding, which took her to the University of Cambridge in England, where she got her PhD in biochemistry. South Africa was fortunate to get you back in 2002, and we were also more fortunate that you returned to your alma mater, UCT. So you've had a great introduction to Wendy's career by the VC. She's gained recognition over the year as over the years as a world leader in viral immunology, recognized for her significant contribution to T-cell immunity in the discipline of infectious diseases and its application to vaccine development and vaccine protection. So her work bridges the field of laboratory science, clinical science, and traditional science. She has over 90 publications to her name, securing large and highly competitive and prestigious both national and international funding. So aside from her research focus, she is a magnificent university citizen, giving back to multiple academic and social events at the university that are aimed to grow and develop the university in important ways. Of note, she was recently appointed as Deputy Director of the Institute for Infectious Disease and Molecular Medicine here in the Faculty of Health Sciences. So Wendy has made many profound contributions in science, but she's also contributed to scientific policy at the highest level. She was a member of the Ministerial Advisory Committee, which we fondly knew as the MAC, on the COVID-19 Vaccine Committee. She embodies the qualities that define a great scientist in person: curiosity, creativity, healthy skepticism, persistence, reliance, integrity, humility, and kindness. And you'll see from some of the quotes from people later, she's also got a great sense of humor, which I think helps. This is all linked to a meaningful contribution to engagement and a relentless focus on transformation and improvement of community health. Her research has focused on the three pandemics: HIV, tuberculosis, and then the novel coronavirus SARS-CoV-2, which caused COVID. She began her career at a pivotal moment in South Africa where the government started to give significant funding to science. And at this time, it was to develop an HIV vaccine, something we're still trying to do, through funding of the South African AIDS Vaccine Initiative. Here she conducted preclinical immunology research, providing critical data that enabled us to take the first HIV vaccine developed in Africa to manufacturing and ultimately to clinical trials in USA and South Africa. Wendy established herself as a lead of an independent research group funded through a prestigious Wellcome Trust Intermediate Fellowship in Public Health and Tropical Medicine. This was followed by a senior fellowship award from the European and Developing Country Clinical Trials Partnership, or otherwise known as EDCTP. She went on to make significant contributions in the TB field. Her career has taken a full circle, however, and she is back now in playing a pivotal role in vaccine development at UCT and has recently founded the Cellular Immunology Platform, which serves as a hub for vaccine evaluation and multiple international programs of excellence. But Wendy is much more than a list of these achievements. So I asked a couple of people for comments. So Catherine Roux, who has worked with Wendy for more than 20 years, I asked her to share some of the flavor of their time working together. I did edit a few of it out. But she does say, working with Wendy has always been a mix of science, laughter, and friendship. The joy of traveling to international conferences and having late night debriefings, usually with wine, and long brainstorming sessions in the lab, each armed with a different colored pen, mapping out ideas, planning research questions, debating experiments. But importantly, Catherine shared us this little story about how Wendy's commitments to science resulted in her breaking down the lockdown rules to write to finish a COVID paper, helped also by a bottle of wine, which we don't know if that was purchased legally or illegally. She didn't say that. So Wendy work in HIV and TB positioned her perfectly to respond to the emergence of the SARS-CoV-2 pandemic. Her group performed high-impact studies describing the strength and duration of immunity to SARS-CoV-2 virus, as well as the virus's ability to evade immunity. These studies generated seminal findings that helped shape policy of the global response to the pandemic. And she's published her COVID work in many high-impact journals, but I think of note was her senior author publication in Nature. So one of her close collaborators who would really have liked to have been here today is Professor Ntobeko Ntuzi, who's the current president of the SAMRC. He was unable to make it, however, he wanted to share a reflection that exemplified her impact and the kind of person that Wendy is. So this is what he wrote. COVID-19 had many lessons for us. One of the enduring ones for me was that we did not have to look beyond our borders for excellence in scientific partnership, a great gift to emanate from the adversity that we were going through was an enduring collaboration with Wendy Burgers and her team. She has a great scientific mind, visionary leadership, and a strong ethical compass, and a wicked sense of humor. It has been nothing but a true pleasure to work alongside her. He goes on to say that Wendy is the doyen of T-cell immunology in this country. She consistently demonstrates an impressive capacity for critical thinking, from the problem solving and collegial leadership, and has brought a level of professionalism and integrity that elevates the performance of our team as a whole. Her ability to engage constructively with complex challenges is admirable. Her commitment to growing the next generation of Black female scientific leaders is unwavering. So beyond her passion, technical expertise, and sound judgment on immunology, Wendy has been an exemplary and one of my favorite collaborators. Her humility, reliability, and willingness to support colleagues at every stage of their career makes her a valuable member of any team. I hold Wendy in the highest regard as a professional and a fellow human being, and I hope I will continue to work with her for decades to come. So in science, we often focus on the metrics, and I think from Intebeko's message, you will recognize that Wendy is much more than metrics. She has a lived experience of a set of values that shape her science and her support for change in this country. This is seen in the staff she employs and the students she recruits and in her teaching. In teaching, she designed a new undergraduate program where she directly addressed transformation in teaching and learning through curriculum change and decolonization. She adopted a new approach to teaching and learning, which firmly rooted her in our context and addressed colonial era exploitation. Wendy is committed and passionate about social justice, both in South Africa and globally. She's an outspoken, and I might add, fearless activist, as evidenced by her support for students during the Fees Must Fall protests, and also more recently, her stand on Gaza. Wendy's journey is purposeful and in science, purposeful in leadership, and purposeful in her contribution to society. She is the type of person we are proud to have in our faculty and university. So I want to conclude my welcome today to share with you a comment from Professor Glenda Gray, the ex-president of the Medical Research Council and the current director of IDORI at WITS. And when I received this email from Glenda, I thought it perfectly captured the multifaceted person that Wendy is. So what Glenda says about Wendy is she is one of the most engaging collaborators I've worked with. I've enjoyed many discourses with her over what a good T-cell vaccine will look like. A lot of these discussions spilled over into evenings to debate, evenings of debate over a glass of wine. Wendy is not only a committed and talented scientist, but she also comes with oodle and passion to match her rigor and concise mind. I've watched Wendy grow as a scientist, and I'm in awe of what she has achieved. Besides science collaboration, I've always admired Wendy's pursuit of social justice, which really makes her the kind of scientist we need in South Africa, rooted in what is right, and what is right and what is the right kind of science that we should be doing in South Africa. Of course, collaboration goes beyond discourse. Sometimes Wendy is my co-conspirator and party partner, and I look forward to many years of science ahead of us, but also to continue to revel in the human that she is. So with that, I'm going to close, and please join me in a warm welcome to Professor Wendy Burgers to the podium for her inaugural lecture. Should do these more often. I feel really great.
Good evening, everyone. Dear friends, family, colleagues, students, and guests, thank you so much all for coming. Thank you, Carolyn, for that introduction. I think you actually gave my whole talk. But also thank you for your kindness and your mentorship over the years. Carolyn has been my head of division, my head of department, and my dean at various times, but so much more than that. You've been such a role model, and you've given me so many opportunities and have really influenced my academic trajectory. So thank you. I'd like to start my lecture with two dedications. The first one is to honor a phenomenal woman, who's my mom, who's sitting here. Mom, you had dreams of your own, but you had to go and work to support your mother and brother at age 18 as soon as you finished school. You dedicated your life to caring for your 5 children, your husband, and your mother, a job that was so often thankless. You didn't get paid, you didn't get promoted, you didn't get leave, and most of the time you weren't valued. If you had had the opportunity to study, you could have been standing here, a professor of botany or ancient history or any one of the subjects you know so much about through your reading. Anything that I've achieved is because of you. You gave me life. You gave me this curiosity and love of learning. The values of hard work, service, empathy, generosity, and integrity. And also a love of reading, of plants, of travel, of the world. Mom, this lecture is dedicated to you. Secondly, I dedicate this lecture to the healthcare workers, academics, and students from Gaza. Like me, you've probably borne witness to the unimaginable horrors on our screens for the last 2 years. I feel like the world has well and truly broken, and I am broken. The Palestinian South African academic Dr. Haidar Eid was recently hosted at UCT. He's a professor at Al-Aqsa University in Gaza, a university that no longer exists. Of Gaza's 12 universities, none is left standing. Israeli forces have perpetrated what has been termed scholasticide, the wholesale, deliberate, and systematic destruction of the Palestinian education system. 993 teachers have been killed, including 193 academics, among them a dean of medicine and a university president. 90,000 university students have not been to class for over 2 years. The numbers coming out of Gaza are hard to contemplate, and of course, they are not numbers, as Palestinian poet and academic Dr. Rifat Alaria reminds us, himself murdered by Israel in December 2023. Professor Eid, in his talk at UCT, spoke about having lost 55 members of his extended family, and he spoke about losing his university colleagues and his students, his brightest students that he had taught over the years, and how they and their young families had been killed by Israel. I could not help but think about my own students, and you, my colleagues, and our university. In addition to all the universities, 33 out of 36 hospitals in Gaza have been completely destroyed. Israel's brutal military campaign has killed 1,700 healthcare workers in Gaza over the past 26 months. Dozens of healthcare workers have been abducted and disappeared. I cannot help but think about you, my colleagues, and our hospital right next door. There is no ceasefire, there is no peace process, and the genocide continues to rage across Gaza and the West Bank, says Craig McIver, former UN Commissioner for Human Rights. I have committed to a full academic boycott of the genocidal state of Israel. As UCT, we have resolved to have no collaborations with academics who are members of the IDF. It is utterly shameful that some funders have withdrawn from UCT, withdrawn funding for student bursaries because they support apartheid Israel. I invite you, my fellow academics in our universities, to commit yourselves to a full academic boycott of Israel as our academic colleagues from Gaza have called us to do, and join efforts with our university leaders in bringing in funders to our university who share our values. If we love justice and work towards justice, let us have the moral courage to stand for justice everywhere. So in my lecture tonight, I hope to share some of the passion for what I've worked on, who I work with, and what we still want to do. I've chosen to tell the story in 3 acts. We begin with the pathogens we cannot see and the wars waged in our bodies without our knowing, where I will share how our immune system records past battles and prepares us for the future ones, and how we harness this memory for vaccines for the parting shot from pandemics past to preparing for those to come, and what we still hope to do, and why the work goes well beyond the lab. Viruses are the unseen enemies. There are 20 billion of them in a grain of salt. They may be unseen, but with advanced technology, we are able to see them. These are electron micrographs of some of the viruses that I've studied, or studied the way our immune system responds to them. Some of them may be familiar to you. Viruses affect every branch of the tree of life: animals, plants, fungi, bacteria, even archaea. So the one in the corner, Acidianus, may not be familiar to you, but for some it may. So my affair with viruses started in high school when I first learned about HIV and decided that was what I wanted to work on. I know some of you have only lived in a world where HIV has been a fact, but then it was a new virus similar to our most recent pandemic, with little knowledge about its biology and no treatment available. The fascination came, and then the horror, from the fact that something so small, unseen, could ravage our bodies, our country, and our world. Other pandemics have come and gone, but HIV remains. 41 million people currently live with HIV. 44 million have died of the virus, and there are still 3,500 new HIV infections every day. As a postdoc, after I finished my PhD, I joined the South African AIDS Vaccine Initiative at UCT and was part of the team led by Annelies Williamson, Carolyn Williamson, and others, developing the first local HIV vaccine based on the subtype of HIV that circulates in South Africa, known as subtype C. We tested in clinical trials, but it didn't mount the protective immune response that was required and was not developed further. My interest turned to what we did not know about the immune response to HIV. In order to design a vaccine in a more rational way, we simply did not know enough about what constituted protective immunity to the virus. The focus of my research was T cells. You all know about antibodies, and when we think of immunity, we think of antibodies, which are made by B cells. They're the first responders on the scene. They can clear out virus and prevent infection altogether. But T cells are important when antibodies fail. They're often overlooked, but they're essential players in antiviral immunity, coordinating the entire immune response. They basically clean up when things go wrong. T cells are having a bit of a moment, for good reason. They just won the Nobel Prize. So regulatory T cells act as brakes of the immune system, preventing it from overreacting, causing autoimmunity or chronic inflammation. They can cure cancer. T cells can kill not only virus-infected cells, but also cancer cells. They can be engineered in the lab, called CAR-Ts, and reinfused to target and kill the cancer. And they can end pandemics, not by stopping infection outright, like antibodies, but by preventing severe disease. But more on that one later. CD8 T cells are specialized cells that can kill virally infected cells. Unlike antibodies that can clear virus floating around in your blood or your airways, T cells kill other cells that send out signals that they're infected. This prevents those cells from becoming viral factories, stopping the infection from spreading further in the body and thus preventing severe disease. T cells also remember past infections, and these are structurally encoded memories. They recognize fragments of a virus presented on the outside of an infected cell and through receptors with exquisite precision produce a deadly strike. Shown here with the T cells in green, readying their poisonous proteins called cytotoxins in red to kill their target cell. And they're serial killers. They patrol the body, migrating through your tissues, and they can last for decades in case you ever encounter the same virus. We study and measure T cells and how they function using a technology called flow cytometry. Here we can determine their specificity, so what part of the virus they target, what type of T cell it is, and what functions they can perform. We tag cells with fluorescent antibodies, so we're using antibodies here as a lab tool that then bind specific markers on or inside T cells. Cells flow past a series of lasers, and when the laser hits the fluorescent tag, they emit light that can be detected and measured using a combination of fluidics, optics, and electronics. Our lab specializes in high-dimensional flow cytometry, and we use this powerful technology to turn the invisible battles of the immune system into visible, measurable data to answer in-depth questions about how T cells respond, remember, and protect us. HIV presents the ultimate vaccine challenge. This is a virus that evolves, hides, and attacks the very immune system meant to fight it, targeting T cells and destroying them progressively until people succumb to infections they can't normally fight off. Our early work, and I'm not going to go through all the data, tried to figure out what T cell responses we need and what defines protective immunity to HIV by studying people living with HIV from very early after infection and tracking those responses over the course of chronic infection and treatment. We performed these studies with Caprisa in Durban in collaboration with Slim and Qureisha Abdul Karim and many others here. Together, our studies showed that specific qualities of CD8 T cell responses, rather than the size or the magnitude of the response, associated with early immune control of HIV. And T cells targeting specific regions of HIV were more important for controlling the virus than other parts. And the lessons learned contributed to our understanding of protective immunity to HIV and the vaccine approaches that are being tested today, which aim to focus the T cell response on those conserved, constrained areas of the virus for more effective responses. In South Africa and across the continent, HIV and TB are intertwined epidemics. TB is an ancient killer, and when it met this new virus on the block, HIV, the results were catastrophic. Your risk of developing TB is 20 times higher if you're infected with HIV. The next years were devoted to studying this group of people at highest risk of TB, to try and glean what features of the immune response are compromised when HIV is there, to tell us about protective immunity to TB. This represents T cells in the real world, where immunity is never tidy, but forged in the overlapping competing demands of co-infection. What we uncovered was even more complexity. We discovered a novel T cell subset that was affected by HIV, a reprogramming of the balance of T cells specific for TB by HIV. And when we examined TB immunity in the lungs of people living with HIV, the site of TB infection and disease, we showed compartmentalization of immune responses, indicating that that's a better place to look, even if those studies are very difficult. So together, these studies contributed some understanding of immune protection to TB for more rational TB vaccine development. Then came a moment when this knowledge became urgently needed worldwide as a new virus emerged to spread into a global pandemic. HIV was not the first pandemic to shape my life or the lives of my family. In 1920, at the tail end of the great influenza pandemic of 1918, my great-grandmother died of flu. She left behind my grandmother and her twin sister to grow up without a mother. That loss has echoed forward through generations. Exactly 100 years after Muriel Dorothy Hamilton died of pandemic influenza, the world was once again in the grip of a global pandemic, this time caused by a novel coronavirus. I spent the next 4 years doing almost nothing else but studying SARS-CoV-2. It felt like history looping back, but this time I was not on the receiving end of the pandemic story. I was part of the scientific response. Having spent much of my career studying how the immune system responds to HIV, I found myself asking the same questions of the novel coronavirus. How does the immune response respond to a new virus? Which parts of the response matter most? And what constitutes protective immunity? Antibodies were shown to be very effective at blocking infection, and the enormous effort to develop COVID vaccines paid off, with the vaccines being shown to be highly effective at protecting people. But another more urgent question arose. What if the virus started to change? And change it did. New variants rapidly emerged. First, a wave of Beta, Delta, Omicron, and the world's attention turned to antibody escape. Would our vaccines still work? Could we stay ahead of the mutations? And the news was not good. The new viruses could potently escape antibodies induced by vaccination or prior infection. But I was drawn to another question. What about the T cells? Did T cells still recognize these new variants? And together with colleagues and collaborators, we set out to answer just that. No making sourdough or pineapple wine for us. During this time, I worked closely with my colleague, Dr. Catherine Ryu, as you heard, a phenomenal scientist. In fact, I regard her as my science soulmate. She left UCT this year, having never received the recognition that she deserved. We achieved so much more working together and produced a body of work demonstrating the effect of viral evolution on T cells, the dynamics and durability of T cell responses, and we looked particularly at SARS-CoV-2 responses in people living with HIV and showed that there was no compromise of the immune response if people were taking their ARVs and were well. We also contributed T cell data to 5 clinical trials of COVID vaccines. What we brought to bear on the problem was our expertise in measuring and characterizing T cell responses, our understanding of how immunity evolves as viruses evolve, and our experience with real-world immunological complexity. Another colleague who I want to honor is the bravest and brightest star among us, South Africa's best scientist, Professor Penny Moore, and she's cringing there. We worked together on all of those same papers too. I have the privilege of calling her my friend and colleague. We spoke on the phone almost every day during the pandemic, in addition to being on 3 or 4 of the same conference calls. Penny, I would not have got through the pandemic without you. Your generosity of collaboration was everything. I want to highlight one of the studies we performed that I'm most proud of, with the major uncertainty regarding the danger that was posed by this newly emerged variant Omicron, identified in late November 2021. We brought together 40 collaborators from across the country and set out to answer that critical question. While the new variant could efficiently escape antibodies induced by vaccination, could T cells still recognize and protect against Omicron? What we pulled off was truly a tour de force. We canceled our holidays, we worked 18 hours a day, 7 days a week to organize all the logistics around sample collection, shipping samples around the country, performing the lab experiments, data analysis, write-up, submission, responding to journal reviews, considerable comments, all in the space of 12 weeks, while dealing with travel bans that delayed critical reagents for our work. What we examined was whether immune responses from people infected or vaccinated. We examined immune responses from people infected or vaccinated, and what we found was really reassuring. Even as Omicron had accumulated almost 40 mutations in its spike protein, T-cell responses remained robust. What we wanted to see was that straight line that you see there. And on average, what we found was there was no change in the T-cell response compared to the ancestral strain. And while these T-cells weren't preventing infection, they were helping protect people from severe disease. And our subsequent studies later showed the same for additional variants, as you can see, the XBB.1 and the hypermutated Omicron variant BA.2.86 that emerged 2 years later. We also demonstrated in our studies the remarkable durability of T-cell responses, with little drop in the magnitude of the response after 2 years, as can be seen there on the slide. Our work garnered significant global interest in the scientific and lay press. We shared regular data updates with the WHO, and sometimes there were 3,000 people on those calls. This was local expertise informing global understanding. We even shared regular data updates with a US team consisting of Tony Fauci, all the officials at the CDC and the White House, who were readying for the US response for the arrival of Omicron. I remember one night in my pajamas at midnight in bed, explaining to a journalist from the New York Times what T-cells were and why they mattered. T-cells made it to New York, London, and Madrid. Science truly doesn't just live in the lab or in scientific journals during a pandemic. It lives in news articles, press briefings, conversations with colleagues, neighbors, and strangers. It lives in translation from the lab bench to the public. Vaccines are almost too good to be true. In the 50 years since the childhood vaccine program, the expanded program on immunization, was introduced by the WHO, it is estimated that 154 million lives have been saved, most of those children under 5 years. A few vaccinations targeting 14 pathogens gained us 9 billion life years. It's dizzying to comprehend that success. The COVID pandemic was a crash course in real-time immunology. Yes, but also in systems of response. It showed us that pandemics are not just biological events. They're social, political, and deeply human. They exposed our vulnerabilities. We learned again that scientific breakthroughs are not enough. In South Africa, we got vaccines late, which cost many lives, and we paid more for them than our European counterparts. Without equitable access to those breakthroughs, science can't protect us. It's not vaccines, after all, that save lives, it's vaccination. But this isn't just about COVID-19 vaccines. Take the new maternal vaccination for respiratory syncytial virus, or RSV. RSV is a major cause of hospitalization in children under 5. Especially those under 6 months. When given to pregnant women late in gestation, it has shown compelling efficacy. Infants born to vaccinated mothers have an 82% lower risk of severe RSV illness in the first 3 months and 70% protection up to 6 months. Yet the cost of this vaccine is staggering. The vaccine would be priced at $200 US dollars per dose, far beyond the reach for wide public health rollout in low and middle income countries. This is precisely why vaccine sovereignty matters. How do we access the tools that protect our most vulnerable if we're reliant on others to decide which lives are worth protecting? The only sustainable way to change this is to develop and manufacture vaccines locally at prices that are affordable. And for the first time, We are building that capacity right here from the ground up. South Africa is now home to two critical players in vaccine sovereignty. Afrigen, the WHO-supported hub pioneering local mRNA vaccine development, and BioVac, a public-private manufacturer leading the charge to produce vaccines on African soil. This is from a few weeks back, the launch of a clinical trial of the first cholera vaccine made completely in South Africa, and that's Glenda Gray vaccinating the first volunteer. So this is all happening against the backdrop of the Africa CDC setting the target for 60% of Africa's vaccines being produced on the continent by 2040 and driving the mechanisms to achieve that. Our lab has partnered with Afrigen to support the development of locally produced mRNA vaccines for RSV, the disease that's so serious in infants. Drawing on our expertise in immunology, we're testing the immunogenicity and protective efficacy of the vaccine in small animal models and setting up the immunological assays that go with that, generating the crucial data needed to move the vaccine into human clinical trials. At the same time, we've joined forces again to develop an mpox mRNA vaccine in response to the recent outbreak that's caused widespread disease across multiple countries on our continent and beyond. Mpox can be devastating, especially for children, pregnant women, and immunocompromised individuals, with painful lesions, long recovery, and in some cases, severe complications. The current lack of access to effective vaccines on the continent make this work all the more urgent. We're working with a number of additional industry partners also, but I want to take a moment to honor Dr. Ryan Keaton, who's also here in the audience, who's leading the preclinical work in our group. She's a scientist with enormous talent, energy, commitment, a phenomenal mentor and trainer, and we couldn't do this work without her deep expertise in experimental models. These collaborations are not just academic exercises, they are acts of sovereignty. They represent not just incremental progress, but a paradigm shift. We now see homegrown entities moving from import dependency to fully integrated vaccine innovation and manufacturing capability. This represents local ownership of the tools we need for our health to protect our people. They're about building science that serves our own communities, because in the next pandemic, and there will be one, we may not be able to afford to wait at the back of the queue again. But it's not just vaccines that we must develop. Someone has to study the pathogens, set up the assays, make the vaccines. It's also the next generation of scientists we must develop. This is our team, many of whom are there sitting at the back. This is a shout out to them, phenomenal young scientists. They're the ones in the lab doing the hard work that I've talked about here. I've also had the privilege of shaping the journeys of the most wonderful young scientists graduating with their PhDs and MScs over the last 15 years. I also want to take the opportunity to briefly mention a number of initiatives I'm leading. Working with industry, we recognize the need to better prepare our students for the reality of industry and innovation in the life sciences. So we've submitted a bid to host the Presidential PhD program in Advanced Biotechnology at UCT. This initiative is designed to equip PhD students not only with cutting-edge scientific expertise, but also with industry-relevant skills, translational experience, and an entrepreneurial mindset, enabling them to drive biotech innovation from lab bench to public benefit. Also, 5 years ago, we launched an undergraduate program in infectious disease and immunology at UCT to lay the foundation for a new major in the science faculty, and have so far introduced 2 half courses as electives. We now aim to evolve this program into infectious disease and innovation. This is my pitch. Integrating innovation and entrepreneurship into the scientific curriculum. By cultivating these mindsets early in the training pipeline, we hope to grow young scientists to become not only knowledge producers, but also problem solvers who make a real impact in our country. And this aligns with UCT's 4th mission of innovation and entrepreneurship. Today, the threats that we've been talking about are not only viral. Vaccines are under attack. There was a massive surge in measles cases in 2023, over 10 million, and over 100,000 people, mostly children younger than 5, died of measles that year, and this from an entirely vaccine-preventable disease. So-called zero-dose children who've not received a single vaccine in their lives are on the rise worldwide. In South Africa, it's estimated there are 280,000 of these children. The dramatic declines in once devastating diseases like diphtheria, pertussis, and measles were so effective that the very memory of those illnesses has faded from public consciousness. Because many of us have never witnessed a child gasping for breath from whooping cough, or the devastation of measles encephalitis, we begin to underestimate the power and the necessity of vaccination. In this vacuum, vaccine hesitancy, fueled by misinformation and disinformation, finds fertile ground. The success of vaccines has not made them less essential, only less visible. But these pathogens are still with us, mutating, adapting, waiting for cracks in our collective defenses. We all need to be vaccine advocates, coming alongside people to hear their fears and providing the facts about vaccine safety. Developing new vaccines is only one part of the solution. There's no point in making vaccines if they won't be used. So while we are doing the science, I call on all of you to help close the gap by building confidence, spreading truth, and championing vaccination. To conclude, we come back to the beginning. If there's one thing that viruses have taught us, it's that we're all connected, biologically, socially, and globally. A virus crossing species in one part of the world can alter lives, economies, and futures across every continent. For me, pandemics are personal and generational. They are part of the stories we inherit and the defenses we try to build, not just in our immune systems, but in our societies. We may not have that HIV vaccine yet, but those decades of research laid the foundation of how we understand viruses, how we craft vaccines, and how we mobilize against new threats that are still to come. The knowledge gained has been repurposed, refined, and redirected, helping us fight SARS-CoV-2 and develop vaccines against pathogens like Mpox and RSV. This is the long arc of science and of a career. Slow, sometimes painful, not at all linear, but always building from one generation to the next. The immune system offers us a metaphor for what good science and good society can be: responsive, adaptable, resilient, and above all, collaborative. Whether through a T-cell targeting an infected cell, or a scientist sharing data, or a student entering the field of immunology, immunity and vaccines are something we build together. And so I return to the question I began with. How do we defend ourselves, all of us, in the age of outbreaks? Not just with science, but with solidarity, as we build a more prepared, more equitable world within the university and without, leading both with scientific excellence and moral courage. These are some of my heroes, inspirations, cheerleaders, incredibly generous mentors and collaborators to whom I'm so grateful. Thank you for your company tonight.
Good evening, everyone. I can't see you.
Professor Burgers, that was quite the tour de force. Thank you for sharing your passion for the work that you do with us tonight and for enlightening us a little bit on T-cell immunity and the complexity thereof. And I hope that in the next few minutes, I'm going to share with our audience some layers of complexity of Professor Wendy Burgers so that you can fully appreciate the awesomeness that is Wendy Burgers, Professor Wendy Burgers. I'm going to say that a lot. At her core, Wendy is a devoted and loving daughter to the original Wendy Burgers. Thank you for blessing us with this courageous and beautiful soul. All your life, you have modeled what it is to be a strong and caring woman through acts of service. And I know that it is a great privilege for Wendy to have an opportunity to take care of you for a change. To the rest of her family, Wendy is a spark of pure joy. We can always rely on Wendy to bring the vibes. As an aangelapte daughter to Madeline and Cedric, she listens with the kindest ears to all their stories. Thanks, Wendy. And as a big sister to Colleen and Dango, she takes a keen interest as she bears witness to their lives. More recently, she received top honors for the coolest auntie from her nieces, Morgan and Cassidy. Teenagers have a way of sussing out fake. And so Wendy is able to be her authentic self at all times, and they really appreciate that. And so to all of you, the family and friends, thank you for being Wendy's joyful distraction around the dinner table, on the couch, on the dance floor. She's a good dancer if you haven't discovered that yet. To all our colleagues here at the faculty, if you don't know this by now, you need to know that Wendy is people soft. She is shaped by working with you every day. So thank you, Mosa, Lionel, Brandon, and all the other members of faculty, especially Kelly and Anati, for hosting us this evening. And thank you, Carolyn, for modeling an example of quiet, unassuming leadership that is very different from what we expect in this environment. And it was how you modeled leadership for us from day one back in 1994. Digby, thank you to you for being a co-conspirator with Wendy as you seek to create an environment in an institute that acknowledges not only the value, but also the contribution of every single student and staff member as equal. And to the members of the Cellular Immunology Platform, we've had this conversation before, do not underestimate each and every single contribution that you make to the study. You all play a critical role, and without one of your contributions, we don't have the paper. So do not underestimate. I know Wendy certainly does not. And then finally, you need to know that Wendy is my brilliant friend. And so now I have to thank myself. Is it all good? Wendy and I first met on Jamie Steps in 1991 during first year. Technically, I met her before she met me. I noticed this bubbly personality that was smiling all the time and very happy, surrounded by people. And I was like, it's first year. What is she smiling about? Just sitting there all on my own, eating my homemade sandwich. Thanks, Mom. And it wasn't until the second semester that Wendy came and joined me in the front row. And I, in retrospect, realized that maybe her first semester results weren't as good as she had hoped. And she was a lot more serious now, but it wasn't too long before we were giggling together in the front row. And we've been on many adventures since, and even now, whether in a board meeting or on a trip somewhere or sitting at a very serious academic lecture, any space is potentially a front row for us. But back to undergrad. This white girl from Littleton Manor high school in Verwoerdburg was making friends with this little colored girl from Retreat. A very unlikely coupling, as Stephen Langtree will tell you, when we walked into the Student Affairs office in 1993 to start the very serious Molecular Biology Society. We felt that it was very important to help our fellow classmates figure out what they're going to do when they finish their undergraduate studies, before they finish their undergraduate studies. And that's incidentally how we discovered this very secret health sciences campus. Thank you, Annalise and Ed, for that. We've gone on many adventures since. We've started the Postdoctoral Fellows Association with Sharon. We've even till today, Wendy is on the board with me. She's the chair of our education NGO Outliers. And it's no wonder that Gay, back in 1994, prophetically coined us les deux. But back to undergrad, those were the early 90s when we were in undergrad. And so there were quite turbulent times in South Africa. A lot of violence across the country, Bishu massacre, the Boipatong massacre, Chris Heine's assassination, Kodesa. And this played out on campus as well with student disruptions and lectures being disrupted. And in 1992, our white friends on campus had to vote to decide whether or not their new Black and colored friends deserved to vote. And I know that this was a very difficult time for Wendy to decide what to do. And we spent a lot of time together as a family discussing things and listening to stories from my parents about exclusion, forced removals, job reservation. And it was at this time that Wendy experienced a formative education in undergraduate studies. It fueled a righteous rage that still burns today. So not surprising when roads must fall and fees must fall happened, Wendy aligned herself with a continued pursuit for decolonization of this university. This is her moral compass. It is unwavering. But I guess on behalf of Wendy, I'd like to thank myself for adding some color to her life. And it would be remiss of me to not end with the quote from the great lyricist Marshall Mathers, with the real Slim Wendy. Please stand up.
Halala, Wendy Burgers, halala. Halala, Professor Burgers, halala. And halala. Viva. Okay, it would be it to steal my thunder, but I want you to conclude that series of halala, just recognizing Wendy Burgers Senior, because in many ways, those are the great halalas of our lives, is the people who bring us to where we need to be. And so I want to just acknowledge both Ken Solomon and Wendy Burgers for being here, because it's not often that we spend this time with family members to see what they do, what our scholars do, and what we get out of them, and what you give up so that they are here. But again, this lecture today has reminded me why we do this, why we gather on the occasion of professorship to say, this is who we are as a people. This is who we are as a community within the faculty. But really powerfully today, Wendy, thank you for reminding us of our commitment and our obligation to interact with our world. Because what did you say? Science does not live in the laboratory. And what a powerful statement to be making, because in so many ways, your lecture reminded us of the location of the biomedical in the psychosocial. And if we don't allow that interaction more freely, we risk the consequences of pandemics through our lives. I was quite struck as you spoke. So as I looked out this evening, I noticed just how many different disciplines of people are here. There are people that I don't always see at lectures by molecular medicine people. And I'm recognizing in that reflection from the room, Wendy, your absolute influence across the faculty. both in affairs of transformation and in your very powerful statement this evening of locating scientists at the heart of social justice. But I also want to remind us this evening that the Western Cape has only a 60% vaccination rate for measles. It is an absolute tragedy that as a world-class university, that's happening on our doorstep. So hopefully we'll recruit Wendy to help us fix that a little bit next year. But let me just close these remarks by just celebrating the nature of hearing our scientists' voices. And every now and again, one comes along like Wendy to kind of locate our science profoundly in the difference we make in the world. But of course, she spoke about companions, I think it was. Did you say companions at the end? And this evening is about companionship and the fact that even this evening we'll all tell stories about the part of the lecture we had. So we invite you after this just to join us for some refreshments, just so that we can continue the storytelling. And so I ask the community to stand as the procession leaves the room.